All right, let's dive right in. Welcome to The Explainer. Today, we are unpacking the fascinating, microscopic building blocks that construct our bodies and quite literally the entire biological world around us. We're talking about amino acids, the absolute fundamental chemical alphabet of life.
To get there, we've got a bit of a roadmap. We'll start with the literal foundations of life, break down the anatomy of these molecules, look at this wild concept called chirality, classify their side chains, and then wrap up by seeing how pH totally transforms them, culminating in the isoelectric point.
Okay, starting off with the incredible foundation of human biochemistry.
When you step back and look at the massive complexity of the human body, from your hair and skin to the enzymes digesting your last meal, it is wild to realize it's all built on a tiny chemical alphabet. Just 20 essential amino acids linked together to form absolutely all of our human proteins.
But here's a stat that's kind of mind-blowing. Out of all the proteins in existence across the entire animal and plant kingdoms, science has really only deeply studied about 5 to 10% of them, and the vast majority of that research is focused almost entirely on human biochemistry. There's this whole universe of protein structures out there in extremophile bacteria and deep-sea creatures that we are just barely beginning to understand.
So let's actually zoom in on that molecular structure. Let's see what makes a single amino acid letter tick.
Every single amino acid shares this exact same core skeleton. Right in the middle, you've got a central alpha carbon. Attached to it, you'll find a protonated amino group, which acts as a base, and a deprotonated carboxylic group, which acts as an acid. Then, to round the whole thing out, there's a simple hydrogen atom and a unique side chain, or R group. That side chain is essentially the biological ID tag. It's what makes every single amino acid distinct from the others.
But you know nature, there is always an exception, right? Enter glycine. Because its side chain is literally just another hydrogen atom, glycine is the only amino acid that lacks a chiral or asymmetrical center. It is perfectly symmetrical, which makes it uniquely flexible when folding into complex proteins. Moving right along, let's talk about chirality, which is basically left versus right-handedness, and how it acts as this incredible biological filter.
Think about your own hands for a second. They're perfect mirror images, but you can't perfectly stack them on top of each other. Well, molecules do this too. So the crucial question is why nature exclusively uses left-handed, or L-amino acids, for our proteins. The secret here lies in water. L-amino acids are highly soluble in water, which is absolutely mandatory since our cells are mostly water. D-amino acids, the right-handed ones, are poorly soluble. In fact, if you try to mix them, they precipitate out. You are only going to find D-amino acids in rare bacterial cell walls or, wildly enough, in the neurotoxic peptides secreted by certain poisonous frogs.
Furthermore, our cellular machinery, our enzymes, they're incredibly specific. They act a lot like bouncers at an exclusive club, actively filtering out incorrect molecular configurations, because only one specific stereoisomer actually has biological utility. An absolute chemical configuration. Every amino acid we use is an S configuration, with just one single exception, cysteine, which is R.
Now, let's look at what I like to call the accent marks of these molecules, their groups and polarities. Amino acids are grouped into families purely based on the chemical polarity and charge of their side chains. First, you have your nonpolar group, loaded with carbon and hydrogen, which actively avoids water. Then you've got polar uncharged, featuring oxygen or sulfur. And finally, you have the electrically charged groups, positive and negative. These specific charges determine exactly how a protein will ultimately fold and interact with its environment. They're like tiny, microscopic magnets. And these side chains? They have some wildly fascinating real-world quirks. For example, the metabolism of tryptophan's indole group literally causes the smell of human feces. Proline has a cyclic structure that is so incredibly awkward to fold, it requires special chaperone proteins just to help it find the right shape. Cysteine features a thiol, or sulfhydro group, that forms super strong bonds essential for giving mammals certain traits like curly hair. And threonine stands alone as the only amino acid rocking two separate chiral carbons. Then you have the modified, non-essential amino acids. They might not be part of the core 20, but man, they do some incredible heavy lifting in your body. 4-hydroxyproline gives your collagen its vital elasticity. Without it, your joints would be completely rigid, but your body needs vitamin C to actually make it. 6N-methylysine goes around repairing your DNA after it gets damaged by UV rays or ultra-processed foods. And gamma-carboxyglutamate acts like a biological claw, binding essential metals like the iron in your hemoglobin.
But what happens when the system breaks down? You've probably noticed a warning on certain juices or vitamin C tablets that says, contains phenylalanine. For a healthy person, totally harmless. But if your body genetically lacks the specific enzyme to metabolize this amino acid, it builds up and becomes highly toxic, leading to severe neurological damage. It's really all about dosage and biology. Think of it like a recent FDA warning about arsenic in a specific batch of chocolate bars. Eating one won't kill you, but eating 54 in one sitting is absolutely a lethal dose. Frequency and biological capability are everything.
Okay, let's shift gears and look at the effect of pH, because these molecules are definitely not static textbook drawings. They dynamically shapeshift based on their environment.
The really crucial thing here is understanding how they react when the surrounding acidity or pH changes. So, rhetorical question, can a single molecule truly act as an acid and a base at the exact same time?
The answer is absolutely yes. Welcome to the concept of the Zwitterion, which comes from the German word for hybrid ion. A Zwitterion is nature's beautiful hybrid. It contains a positive charge on its amino group and a negative charge on its carboxyl group, which results in a net electrical charge of 0. This unique state is exactly what allows the molecule to remain perfectly soluble and functionally active in the watery environment of our cells.
If you track it through the pH scale, the amino acid behaves incredibly dynamically. In a highly acidic environment with low pH, like inside your stomach, it acts as an acid holding on to protons and carrying a net positive charge. At a neutral point, it reaches that perfect Zwitterion state. And in a highly basic environment with high pH, it actually sheds those protons, acting as a base and taking on a net negative charge. It sequentially loses protons first from the carboxyl group and then from the amino group.
Which perfectly tees up our final concept, finding exactly where that perfect hybrid balance happens. We call this the isoelectric point. Now, a quick heads up on the word neutral here, because it can be tricky. Remember, the neutrality of an amino acid is about its internal electrical charges canceling each other out to 0. It does not necessarily mean this happens at a neutral pH of 7 on the standard acidic basic scale. Because every amino acid has completely unique chemical properties, we can actually mathematically pinpoint its perfect balance by averaging its two dissociation constants, or pKa, values. Take alanine, for example. We take its first pKa of 2.35, add its second pKa of 9.87, and divide by 2. This simple math tells us that at exactly 6.11 pH, alanine reaches its peak Zwitteron form.
And those individual pKa values are absolutely vital. They indicate the exact pH where 50% of a specific group is ionized. Inside our bodies, these act as critical biological buffer zones. When our systems experience sudden, massive acidic or basic shifts, these amino acids actively absorb or release protons, literally fighting to keep the pH stable. This is exactly how they keep our essential proteins from breaking down when our biological environment fluctuates.
So we've basically mastered the individual letters of this chemical alphabet today. We've seen their anatomy, their quirks, and their dynamic, shape-shifting nature. But here is the ultimate question to leave you chewing on. Now that you know the building blocks, what mysterious genetic instructions sequence them together into the elastic spider silk or the oxygen-carrying hemoglobin that keeps the whole world alive? That is the incredible mystery of proteins, waiting to be decoded. Thanks for joining me on The Explainer.